Hi, my name is Mathieu Quesnel-Vallieres. I am a postdoc in the Barash, Lynch, and Thomas Tikkonen Labs at the University of Pennsylvania. Today, the work that I'm presenting was conducted in close collaboration with Anupama Jha, a former grad student in the Barash lab. I am happy to report our findings on this project, which aim to draft a molecular definition of cancer by interpreting deep learning models trained with transcriptomic data. The motivation behind the project was quite simple. If you look at the textbook definition of cancer, you get a rather vague definition that says that cancer is a collection of related diseases that share common features, including how cells start dividing without stopping and then invade surrounding tissues, thereby disrupting their function. How these cells can ignore signals that tell normal cells to stop dividing. And there's also an important genetic component. That is that cancer is essentially a genetic disease that is caused by mutations or genomic alterations in genes that control the way cells grow and divide. And this definition is quite on point. Taking the Cancer Genome Atlas, or TCGA, 89% of the samples in this data set have at least one mutation in one of the 9 cellular pathways I'm showing here, which all ultimately control the way cells grow and divide. Now, the question that we had was whether these mutations in different cancer pathways converge on common transcriptomic variations. And we also wanted to know whether there are additional non-genetic determinants of cancer. The objective here is to find transcriptome signatures defining cancer state through an interpretable deep learning network that classifies normal versus cancer and assess the impact of these signatures on cell biology. At the high level, what we did was to take a large number of RNA-seq samples, quantify transcriptomic features, train a model that classifies sample between healthy and tumor. Once we have a successful model, we want to interpret this model to figure out which transcriptomic features have the greatest weight in the model and use these features as a transcriptomic definition of cancer, from which we can assess whether we mostly recapitulate the known genomic signatures of cancer or unveil new transcriptome-specific patterns that would inform us on the biology of cancer. In more details, and using a graphical representation of our method, we first use transcriptomic features, let's say, for example, protein coding gene expression data. We reduce dimensionality with an autoencoder, and then train a feed-forward network that classifies samples between cancer and healthy tissues. Once we have a successful model, we interpret the model using enhanced integrated gradients, which is a method that our group published and reported last year in this paper referenced here. And enhanced integrated gradients allow us to attribute an attribution value to each of the features that we feed into our model. So taking the case of protein coding gene expression, every single gene that we quantified and fed into the model will get its own attribution value. And here, the higher the attribution value, the more weight this feature has in how the model determines whether it is dealing with a cancer or a healthy tissue. Of course, in order to train models that perform well, we need a large number of samples in our training set. Here I show the tissues and cancer types that are represented in our training set on the x-axis, along with the number of samples for each tissue or tumor type on the y-axis. There's a large number of these samples that come from TCGA and GTEx, and one of the big hurdles that we faced was to assemble a data set that would allow us to overcome how TCGA and GTEx are perfectly confounded. That is that all GTEx samples are healthy samples and almost all TCGA samples are cancer samples. So in order for us to avoid predicting technical biases inherent to the use of these two large data sets and make sure that we were modeling cancer versus healthy rather than GTEx versus TCGA, we included 12 smaller data sets, some of them that included paired healthy and tumor tissues from the same donors. And we also performed a tissue-specific mean correction of healthy and tumor samples across the 14 data sets. Another important step in our approach was to process all the samples in the exact same way, starting from FASTQ files. That means that we actually used legacy TCGA. And this was an essential step to reduce the technical biases that we were introducing in our data set as much as possible. Now I'm going to present results from the first step, which is the training of a model using expression data from close to 20,000 protein coding genes. We were able to train a model that has accuracy, precision, and recall between 94 and 96%. Importantly, the accuracy is high across the data sets despite class imbalance. If you look at the number of samples in each of these data sets, you can see that the numbers vary widely. And the fact that the accuracy remains high across the data sets indicates that our model is predicting classes based on biological signal and not confounding factor. So that's great news. We have a model that works, and that tells us that there exists a protein coding gene expression cancer signature. But before we moved on and started interpreting our model and investigating the biology, we wanted to know if there were other types of transcriptomic features that could similarly be used to distinguish between healthy and cancer tissues. So are there other transcriptomic signatures of cancer? There's all kinds of information that we can extract from RNA-seq data. Very often people use RNA-seq to quantify gene expression of protein coding genes like we just did, but one can also get information about the expression of long non-coding RNAs or about splicing patterns. Finding that it was possible to train a model to distinguish healthy from cancer using protein coding expression was great, but it wasn't so surprising given that it has been known for quite some time now that tissues have clear and specific signatures of protein coding gene expression, which is something that's not very clear about long non-coding RNA expression and splice site usage, especially when comparing cancer to a healthy tissue. And so the question that we had was, are there long non-coding RNA expression and splice junction usage signatures in cancer? And we discovered that yes, indeed, as evidenced by the high accuracy, precision, and recall of our models trained with long non-coding RNA gene expression or splice junction usage, and again, high accuracy across data sets, that there exists long non-coding RNA expression and splice junction usage cancer signatures. Next, we were really interested in knowing how these newly identified cancer signatures compare to known molecular features of cancer. So we turned to the interpretation of our models using enhanced integrated gradients and generated attribution values for each of the genes and splice junctions that we trained our models with. We established a list of high attribution features using cutoffs that are described here. And this gave us a list of 1,053 genes that are generally more highly expressed in cancer and 715 protein coding genes that have lower expression in cancer. Similarly, we have 852 long non-coding RNAs that are more highly expressed in cancer and 911 genes that have lower expression in cancer. And we have a set of 562 splice junctions that are enriched in cancer. Now, because the selection of these high attribution values was based off of median values that were calculated over all of the samples of all the cancer types, the first thing that we did was to evaluate how consistently enriched or depleted these features are across cancer types. Here we have 100 genes that have the highest attribution values, either negative, so that means that these are the most depleted genes in cancer, or positive, so the most enriched genes in cancer. And each gene is broken down into 14 data points, which correspond to the 14 cancer types that we tested. And you can see how these 14 points are tightly grouped for this gene here and how this is true for the 100 depleted genes and the 100 enriched genes. And this tells us that if a gene has a high attribution value in any cancer type, it also has a high attribution value in the 13 other cancer types that we tested. And this shows that the transcriptome signatures of cancer that we identify are consistent across cancer types. I'm only showing data for protein coding genes, but this is also true for long non-coding RNA genes, as well as splice junctions are enriched in cancer. I'm showing for reference genes from the catalog of somatic mutations in cancer that are annotated as oncogenes or tumor suppressor genes, just to show how these genes that are known oncogenes and tumor suppressors actually have smaller attribution values in general than our top high attribution genes. In fact, most of the high attribution genes that we find are not labeled as oncogenes or tumor suppressor genes in the cosmic database. If you look at the overlap here between genes that we find with high positive attribution values or genes that are enriched in cancer, and the overlap with cosmic tumor suppressor genes or oncogene, you can see that the overlap is very minimal. It's also true for genes that are depleted in cancer. So this indicates that we uncovered new molecular components characterizing cancer state. If the genes that make up our transcriptomic definition of cancer differ from known oncogenes and tumor suppressor genes, is it also true for their functions? This is a map showing Goterms from biological functions that are significantly enriched from genes that are enriched or depleted by expression or splice junctions in cancer. Each node is a Goterm, and edges show terms that have genes in common, with the thickness of the edge representing the number of genes that two terms have in common. You can clearly see how genes that are depleted in cancer and shown in blue form their own cluster here on the right, and how the functions that are represented are related to traditional definition of cancer with transcriptome transcription-related functions and genome integrity-related functions with chromatin regulation and histone modification. On the other hand, you have here on the left and in red, genes that are enriched by expression or splice junctions that have a lot to do with RNA processing. And this is very interesting because disruption of RNA processing is currently emerging as an important component of cancer biology. Finally, an important question that remained was whether the genes that we find to be enriched or depleted in our models participate in the onset and or maintenance of cancer, or whether the expression and splicing changes that we observe are secondary manifestations of the loss of driver genes and are therefore more like markers of established cancer. We did some functional analyses to start addressing this question, and we found that genes that are enriched or depleted in cancer are more evolutionarily conserved in a set of neutral genes, and these neutral genes would have similar expression in cancer or healthy tissue, and we use this set as a reference. I'm only showing data for protein coding genes, but this is also true for long non-coding RNA genes, as well as genes that have splice junctions enriched in cancer. So these are also more evolutionarily conserved than their neutral set. We also find that these genes are under much stronger selective pressure against loss of function mutations in the human population relative to the neutral set. And this is measured by the Nomad left score, which is a representation of how often a gene displays a loss of function mutation in the human population. So the lower the left score, the more selective pressure against loss of function. And again, this is only for protein coding genes enriched by expression, but this is also true for long non-coding RNA genes, as well as genes with splice junctions enriched in cancer. And last, we observed that long non-coding RNAs that are enriched or depleted in cancer are much longer in terms of transcript length than the neutral set. And they also have a higher picnon density, shown here on the right. If you're not familiar with picnons, these are 16-mer intergenic and intronic DNA motifs that are sometimes differentially transcribed in some cancer types, and that can also affect the oncogenic properties of long non-coding RNAs. So together, these data indicate that at least some of the genes that we identify as being enriched or depleted in cancer, either by expression or splice junction usage, might have essential functions in cancer cells. As this talk comes to a conclusion, I want to highlight how in a network analysis, genes that are enriched in cancer by expression or splicing and that have functions in RNA processing and that are shown over this map in the red nodes how these genes are tightly interconnected with the cancer pathways identified from genomic studies and that I mentioned in the introduction. And this observation makes it tempting to suggest that RNA processing is actually a molecular process on which several other cellular pathways converge in cancer. To summarize, we identified protein coding gene expression, long non-coding RNA expression, and splice junction usage signatures of cancer that differ from the genetic definition of cancer. We found that genes with lower expression in cancer are associated with a traditional view of cancer with roles in transcription, DNA regulation, and genome integrity. On the other hand, we found that genes with higher expression in cancer are associated with functions related to RNA splicing and processing, which is an emerging theme in cancer research. And finally, the fact that we observe a high conservation and selective pressure against loss of function in genes with high attribution values by expression or splicing suggests that at least some of these genes that we identified have essential functions in cancer cells. I thank my mentors, Dr. Josef Barash, Dr. Kristen Lynch, and Dr. Andrei Tomas-Tikhonenko for their feedback and support. It was a real pleasure working on this project with Dr. Anupama Jha, who performed all of the computational modeling and the interpretation for this project. I thank you for your attention.